Let's go over the answers to the warm-up review. First question, which medication overdose is treated with sodium bicarbonate? So you use bicarb to alkalinize the urine, which is going to trap weak acids. So you give bicarb to treat overdose with weak acids, such as aspirin. Next, a patient presents with tinnitus, dizziness, headaches, and GI distress. So what drug is potentially causing these symptoms? Well, this syndrome is called synchonism, which is typically caused by either quinine or quinidine. And the last one, which testicular tumor is associated with each of the following statements? Composed of cytotrophoblasts and syncytiotrophoblasts is choriocarcinoma. May present initially with gynecomastia is Leydig cell tumors, or rarely you can get a Sertoli cell tumor that can cause this as well. Elevated AFP would be a yolk sac tumor primarily, but you also need to think about embryonal cell carcinoma. Elevated beta HCG is either choriocarcinoma or embryonal carcinoma. And finally, histologic appearance similar to chondrocytes with cytoplasmic clearing is a seminoma. Now let's get started with the lecture.
Hello, and welcome to our Step 1 video on bacterial basics. I'm Dr. Jennifer Shuford, and I'm an infectious disease specialist. I'll be giving some lectures in the microbiology course. In this lecture, and in the next few lectures, we'll be discussing bacteria. In this lecture, we'll discuss bacterial structures, staining, and genetics. Let's start with bacterial structures. This can be a tedious subject, but it's still important and fair game on your test. So let's start with the cell membrane. Now look at this image of two different bacteria. The gram-positive organism is on one side and the gram-negative on the other. The plasma membrane is at the bottom of each of the figures. The plasma membrane is composed of a phospholipid bilayer that helps regulate what goes in and out of the cell. Some substances can diffuse across the membrane, but charged molecules usually need a transport protein or a protein channel to traverse that membrane. If you look above the cell membrane, you'll notice that the two figures look very different. The gram-positive organism has a thick peptidoglycan layer, also referred to as a cell wall. A cell wall lends support to the cellular structure and can protect against osmotic changes. Peptidoglycan contains polysaccharide chains that are cross-linked by peptides. The peptidoglycan layer dictates how much crystal violet stain is taken up during the gram stain procedure. In bacteria with thick peptidoglycan layer, the crystal violet stain is taken up well. The bacterium appears purple under the microscope, and the bacterium is referred to as gram-positive. Bacteria with a thin peptidoglycan layer and an outer membrane are unable to retain that crystal violet during the decolorization phase, and instead they take up the counter stain and therefore appear pink under the microscope at the end of the gram stain procedure. These are called gram-negative bacteria. These structural differences do not merely impact our staining studies, but also determine other interactions between the bacteria and the host. Let me give you an example. The thick peptidoglycan layer of the gram-positive organisms contains lipoteichoic acids, but gram-negative organisms don't have these acids in their structure. The lipoteichoic acids in the cell wall induce cytokine production, which then sets off a number of reactions within the host. Just to remind you, acute phase cytokines include interleukin-1, interleukin-6, TNF-alpha. We'll talk more about cytokines in immunology, but I wanted to review those quickly. Now look at the other figure, the one of the gram-negative bacterium. What you will notice on the gram-negative figure, besides the very small peptidoglycan layer, is the presence of an outer membrane. This is a distinctive feature of the gram-negative bacteria. Again, this structural difference impacts how it interacts with the host. The outer membrane contains something called lipopolysaccharide, or LPS. LPS is an endotoxin that's a potent stimulator of cytokines. So here we see that the gram-negative bacteria can stimulate the acute phase cytokines too. Gram-negatives do not use lipoteichoic acid like gram-positives, but rather the LPS contained within the outer membrane. The inflammatory reaction from LPS can result in septic shock, and it's a huge contributor to the morbidity and mortality seen in severe gram-negative infections. Let's continue to look at that figure of the gram-negative bacterium. The gram-negative organism has both an inner and an outer membrane. In between these two membranes is the periplasmic space. This periplasmic space is the location of many proteins, including beta-lactamases. What are beta-lactamases? These are enzymes that break down beta-lactam antibiotics like penicillins and cephalosporins. So gram-negative bacteria can carry around a resistance mechanism, a beta-lactamase enzyme, in their periplasmic space. Okay, let's look at the surface of the cell and talk about the fimbriae. These are hair-like structures that mediate adherence of bacteria to surfaces, including the surfaces of other cells. These fimbriae are what allow bacteria to colonize membranes throughout the body, like the respiratory tract or the GI tract. Now, similar to fimbriae are pili, or pilus in its singular form. The pilus forms an attachment between two bacteria during conjugation. And what is conjugation? Remember it like this. Conjugation describes a bacterial conjugal visit. A pilus allows transfer of DNA from one bacterium to another. These pili used during conjugation are sometimes called sex pili or F pili. And now flagella. Flagella are usually longer than fimbriae and pili. They're filamentous structures that whip around, providing mobility to bacteria. Now let's go through a few more bacterial structures. Let's stay on the surface of the cell and talk about glycocalyx. Glycocalyx literally means sugar coat. It refers to a gelatinous polysaccharide coating for bacteria. It aids in attachment to foreign material like indwelling catheters, as well as natural surfaces like teeth. If the glycocalyx is organized and firmly adherent to the bacterium, it's called a capsule. If it's loosely adherent with less organized structure, it's referred to as slime or biofilm. Let's take a second to talk about capsules. Not all bacteria have capsules, but many bacteria that cause significant human disease do. A capsule protects the bacterium against phagocytosis, allowing it to elude the immune system just a little longer than some other bacteria and giving it time to divide and therefore cause overwhelming infection. A capsule can be considered a virulence factor. A virulence factor is some characteristic of an infecting organism that makes it stronger or more virulent. So how can we tell if a bacterium has a capsule? Well, one way to tell is through the Quelling reaction. An anticapsular serum can be added to the bacteria. If there's a capsule, it becomes swollen, looking under a microscope, and this is called the Quelling reaction. One way to remember this is Quelling equals capsular swelling. So I want you to remember the Quelling reaction. But I'm also going to give you a list of some of the encapsulated organisms. A mnemonic you can use is even some pretty nasty killers have shiny bodies. This is to help you remember these organisms. E. coli, just some strains, but E. coli, Streptococcus pneumoniae, Pseudomonas aeruginosa, Neisseria meningitidis, Klebsiella pneumoniae, Haemophilus influenza type B, Salmonella typhi, group B strep. So even some pretty nasty killers have shiny bodies. Of that list, I think that 3 are especially important and are tested routinely. These are Streptococcus pneumoniae, Haemophilus influenzae, and Neisseria meningitidis. These can be remembered by the word SHIN. Strep pneumo, Haemophilus influenzae, and Neisseria meningitidis. These are big-time players. They can cause overwhelming sepsis and death in splenectomy patients who are unfortunate enough to be infected. But there are also effective vaccines against all 3 of these organisms. A splenic patient should always be vaccinated against these bacteria. The capsules of these bacteria, which serve as virulence factors for the bacteria, can also serve as antigens for vaccines. So Pneumovax, a pneumococcal vaccine, is derived from the capsular polysaccharide, as are vaccines against Neisseria meningitidis and Haemophilus influenzae. One other important fact regarding capsules, Cryptococcus neoformans, which is a yeast, also has a capsule. Cryptococcus causes meningitis in AIDS patients and has a capsule that can be easily identified using India ink stain. So when you think of encapsulated organisms, Cryptococcus should be high on that list, even though it's not a bacterium. All right, so just a second ago, I mentioned something called slime. Slime, or biofilm, is the polysaccharide that is produced by some bacteria and that can cover a surface of an entire microcolony of bacteria. When bacteria are under this covering, they're usually more resistant to antibiotics and more difficult to remove from surfaces. Okay, let's move inward from the surface of the bacterium and talk about some of the intracellular structures, starting from the ribosome. Ribosomes are where proteins are synthesized. 2 subunits fit together and work to translate mRNA. Bacteria are prokaryotes that have a 50S and a 30S subunit, which come together to make a 70S ribosome. Eukaryotes, like us, have a 60S and a 40S ribosome subunit that come together to make an 80S ribosome. The S refers to sedimentation coefficient. That is a measure of how rapidly these ribosomes sediment out in a solution. So don't spend too much time trying to get these numbers to add up. They don't. The reason I'm telling you this about the ribosomes of prokaryotes versus eukaryotes, though, is to highlight that they have different ribosomes. This structural difference provides a very useful target for antibiotics because we can preferentially target bacteria and not host cells. We'll talk about such antibiotics in another part of the course. Now, plasmids. Plasmids are pieces of genetic material that are separate from the chromosomal DNA. They replicate separately from the chromosomal DNA. Plasmids often contain genes that may be helpful to bacteria in certain situations, but that aren't essential for their survival. For example, plasmids can carry genes for antibiotic resistance or for toxin production. These plasmids are pieces of DNA that can be transferred from one bacterium to another during conjugation. So conjugation allows the transfer of resistance and virulence factors from one organism to another by way of plasmids. Okay, so besides conjugation, plasmids, what are some other mechanisms of genetic change in bacteria? Well, let's talk about 3 others. First, transformation. Transformation occurs when DNA is released from a lysed cell and is then taken up by a living bacterium. These DNA fragments can then be incorporated into the chromosomal DNA of the recipient bacterium, resulting in a recombinant bacterium. The recipient bacterium now has a recombination of its original chromosomal genes with those from the acquired DNA fragments. Transformation is only an option for a few species of bacteria. Interestingly, these include those Shin organisms that we just talked about a few minutes ago: Strep pneumo, Haemophilus influenzae, and Neisseria meningitidis. Now let's discuss transposons. These are small segments of DNA that can self-excise and then relocate to another place on the chromosome. They can also relocate to a different chromosome altogether or to a plasmid. Some transposons are very small and carry minimal information. Others can carry antibiotic resistance or virulence factors. So how do you think that transposons could be responsible for spreading antibiotic resistance? Well, if the transposon was relocated from a chromosome to a plasmid, you can see how that gene coding for antibiotic resistance could then be spread to other bacteria via conjugation. Lastly, let's discuss transduction. Transduction relies on something called bacteriophage, or just a phage. Phages are viruses that infect bacteria. The phage attaches to the bacterium, injects its DNA into the cell. More phage DNA and phage proteins are made and are repackaged into new viral capsids. These are then released when the bacteria ruptures. What's important to know here is that phage replication isn't a clean process. Some of the bacterial DNA can end up inside that viral capsid. So when that bacteriophage goes to inject its own DNA into the next bacterium, what it's actually injecting is both viral and bacterial DNA. The bacterial DNA can then be recombined with the DNA of the infected cell. Okay, that's about it for general structure and genetics of bacteria. Now let's talk about some bacteria that have unusual characteristics and how you identify these. There are 2 bacteria that I want you to remember because they have unusual cell membranes or walls. And the first is Mycoplasma species. Unlike most other bacteria, Mycoplasma requires sterols for their plasma membrane, and they have no cell wall. Why would that be important that they have no cell wall? Well, one reason is that many of our antibiotics target the cell wall. These would not be very useful against Mycoplasma. Another bacteria I want you to remember is Mycobacteria species. Mycobacteria have cell walls that contain mycolic acid and are rich in lipids, making them somewhat resistant to gram staining, but very resilient in a variety of environments. All right, we've been touching on this the whole lecture through, but let's focus on staining for a moment. As we already mentioned, gram staining divides most bacteria into one of 2 groups, gram-positives or gram-negatives. Gram-positive organisms stain blue or purple because they retain the crystal violet primary stain. Gram-negative bacteria are going to stain pink or red because they lose the crystal violet during decolorization and they retain the counter stain. Now, there are some bacteria that just don't gram stain very well, and I'll go through a few of the important ones right now. You'll recognize a couple of these. So mycobacteria. Remember that mycobacteria have an unusual cell wall with mycolic acids and high lipid content, and it causes irregular uptake of gram stain. And mycoplasma. Do you remember? We just talked about this. Remember, mycoplasma has no cell wall, which affects its staining. Treponema. Now, treponema are gram-negative organisms that are shaped like very tiny corkscrews, but they're difficult to visualize because of their size, and they can be seen better with dark field microscopy. Legionella pneumophila. Legionella stains poorly because of unusual branched chain fatty acids that they have in their cell wall. We've talked about some of the limitations of gram staining now, so let's talk about some other important stains that are used to identify bacteria. There's Giemsa stain. Giemsa can be used to stain Borrelia, Rickettsia, Chlamydia, Plasmodium, Trypanosomes, and Histoplasma. Then there's PAS, periodic acid shift stain. PAS stains glycogen and mucopolysaccharides. It can be used to diagnose Whipple's disease caused by Trophirima whippleyi. Zeal-Nielsen stain. The Zeal-Nielsen is used to stain acid-fast organisms like Mycobacterium tuberculosis. And India ink. We mentioned this when we were discussing capsules, but this stain, it isn't routinely used for bacteria, but it's important for fungus. And remember, this is for Cryptococcus neoformans. Now, silver stain. Silver stain is used for fungi, including Pneumocystis, and it can also be used for Legionella. And just so you know, being familiar with these few stains can really pay off for you on the test. Now it's time for your end of session quiz, so I want you to pause your video, go through those questions now, and then restart your video so we can go through them together. All right, first question. What bacterial structure has the following function? It mediates adherence of bacteria to the surface of a cell. That's the fimbriae. Protects against phagocytosis. That's capsule. Provides rigid support to the bacterial cell and protects against osmotic pressure differences. This describes the peptidoglycan layer of the cell wall. Space between the inner and outer cellular membranes in gram-negative bacteria. This is the periplasmic space. Motility. Motility is mediated by flagella. Bacterial form which provides resistance to dehydration, heat, and chemicals. This is a spore. Forms attachment between two bacteria during conjugation. This is the pilus, sometimes called the F pilus or the sex pilus. And independent pieces of genetic material within bacteria that may contain genes for antibiotic resistance. This describes a plasmid. Okay, next. What stain is required to see the following organism or substance? For Cryptococcus, you can use India ink. For PCP or Pneumocystis, you can use silver stain. For chlamydia, you can use a Giemsa stain. Next, which organisms are not well visualized with gram stain? Treponema. Remember that treponema have a thin corkscrew shape that's too thin to be well visualized with a gram stain. Legionella. Legionella's cell wall has unusual fatty acids that resist stain. Mycoplasma species, like Mycoplasma pneumoniae, they have no cell wall, so they don't gram stain. And Mycobacteria, they have a high lipid content that resists gram staining. Next, which bacteria are encapsulated? The mnemonic to remember is even some pretty nasty killers have shiny bodies. So that's some strains of E. coli, Strep pneumoniae, Pseudomonas aeruginosa, Neisseria meningitidis, Klebsiella pneumoniae, Haemophilus influenzae, Salmonella typhi, and Group B strep. And then it's not a bacterium, but another important pathogen to remember that has a capsule is Cryptococcus neoformans, which is a fungus. And last question, by what method are plasmids exchanged between bacteria? This is called conjugation. And remember that this process utilizes a pilus. And that's it for our end of session quiz, and that's the end of our bacterial basics video. I'll see you again soon.